I'll start by really considering a typical NGS workflow, right? You could see that Agilent really focuses on the two flanking portions of the workflow, both the upfront sample prep and library prep, as well as the downstream bioinformatics. So to begin, I really want to focus on an aspect of the workflow that's really near and dear to the hearts of everyone at Agilent Genomics, and that's targeted enrichment. So I think it's fair to say that the cornerstone of Agilent NGS portfolio is the SureSelect technology, specifically for targeted enrichment. And of course, targeted enrichment is being able to specifically pull out the library fragments that you are interested in understanding and interrogating, and moving those along to sequencing rather than relying on whole genome sequencing approaches. And of course, Agilent is the originator of the targeted enrichment approach, which goes back to our origins as part of Hewlett-Packard. And then HP actually designed a process in which up to 3 million unique oligo sequences could be printed in a single run. And these 120 base pair long biotinylated RNA baits are then used in our reaction to hybridize tightly to these target DNA NGS libraries, allowing you to enrich those targets and interrogate them, of course, by sequencing. So Agilent is not only the first, but also the most broadly adopted and widely published target enrichment platform in the world. But we don't want to we don't want to remain content. We want to continue to invest in our portfolio and really benefit our customers with some of the more emerging applications that are starting to come out. In fact, we recently completed a major production and performance improvement project that allowed us to bring the Exome V8 panel to market. And we'll talk a little bit about some of those improvements as well as the Exome V8 more specifically in later slides. But before we get there, I do think it's worth noting that our robust manufacturing platform and world-class quality process has continued to enable a lot of our clinical partners. This includes the first NGS-based IVD cancer test in China, the first ever NGS-based IVD cancer profiling assay in Japan, and many other regulatory approved tests in Europe and in the US. So I want to briefly go over some of the improvements that I spoke about before and really focus on 4 key areas. First off, we've improved our in silico design process that is responsible for translating targeted regions from the genome or the transcriptome into complementary probe sequences. Each probe sequence needs to be optimized to ensure that these specific targets or regions of interest still work alongside the tens to thousands of other probe sets that you might see in your panel that make up the panel. And as you can imagine, this is no simple feat. So by utilizing innovative machine learning processes, we've made this design process, not only superb today, but capable of continuing to get better as we repeat the process time after time. Secondly, we've adopted a major innovation in terms of oligonucleotide printing, whereby we now synthesize against both the forward and reverse strands of a specific target region. This can reduce rare, but sometimes troublesome artifacts that historically have been really difficult to enrich for. And then third, we came up with a new process of probe recovery after the printing process, during which the DNA oligos are released from the synthesis substrate and collected into a common pool. So this improved probe recovery process is particularly relevant for those really tricky GC-rich regions that you tend to find in clinically relevant genes or conserved genes. Finally, fourth, we've been able to adopt all these cool key innovations that I just mentioned without altering the conversion process that transforms DNA oligos that we print into biotinylated RNA baits. So essentially, we've made all these key improvements without impacting or negatively affecting the RNA baits themselves. I'd like to take a minute just to focus on the new machine learning probe selection process and why we're so excited about it. So just a little bit of history here. SureSelect probes in the past would have been designed by tiling across the genomic region of interest using a series of overlapping 120 base pair sequences that you see in that cartoon image on the left, right? And as you can see in that image, this typically leads to a suboptimal design, as those probes will go past your region of interest and cause these little overhangs that are flanking your target region. So with our new process, what we're able to do is we can virtually test a much larger number of possible probe designs and select those that are most likely to be effective and enrich only the regions of interest that you're actually interested in, right? So a lot of those overhangs and that you've seen in that previous design, we can essentially eliminate those. Ultimately, this machine learning approach is leveraging learnings from a decade of experience in making enrichment panels, which is why Agilent's unique background has made this approach really the most robust in the industry. Okay, now all that sounds great in theory, but let's take a look at what this really means in terms of performance. And I have a couple of graphs and slides here comparing our legacy process to our new and improved process for you guys to kind of take a look at here. So first off, you can see that in this particular example of a region of interest that's about 4.5 megabases, this is a custom panel. When you actually do the design previously, it turns out to be a little bit larger footprint, about 6.8 megabases. But what this new process does is it brings it down to a slimmer 5.4 megabase panel. So that means that we've done away with over a megabase of unwanted flanking sequences without increasing zero coverage targets at all, which you see in that last graph there, right? In fact, it actually went down with our improved process. So all this translates into reduced sequencing costs, fewer orthogonal validations, and you can all imagine how much time and dollar saves this essentially adds up to. So just making things a little bit more efficient. Beyond that, if we stick with the same panel and zoom into a few regions using the Integrated Genome Viewer, or IGV, to take a look at some of these read pileups, we can see that there's increased uniformity across the panel, and this translates to better coverage of GC-rich exons in clinically relevant genes like ARX here. And essentially, for those that are not familiar with this view, and hopefully you can see my pointer, the legacy track is now then compared with this improved track as well, right? Same thing for other genes like BRAF. And one of the things that I'd like to really have you guys keep in mind here is that when you're looking at these comparisons, keep in mind that this is using the same sequencing amount for both the legacy and improved. So you're not just seeing a higher read depth with regards to the improved, you're actually seeing a more efficient use of each sequencing run. So again, going back to that efficiency. So today I'm really excited to say that for the first time ever, we've utilized our new and improved bait production process to produce a brand new SureSelect human all exome panel, which we're calling version 8. And not only does this panel include all the innovations that I just spoke about, it also offers a sleeker design with expanded pre-design and custom add-on content if you want to interrogate non-exonic regions of the genome, such as UTRs and other non-coding regulatory regions. As you'd imagine, the performance characteristics are excellent on this panel, and we certainly have white papers and data sheets available on our website for you guys to consider. And this also fits seamlessly into our NGS workflow, which you will see a little bit more of as we continue along in this presentation. Just a couple of more points on the human all exon V8 panel. It still gives you the same reliable, comprehensive content that you're used to on the version 7. But it actually reduces the design size, and again, making it a little bit more of a sleeker design by 20%. It has the same high on-target rate as you've seen previously in version 7, 10% higher 30x coverage, and also a 30% reduction in fold 80 penalty. So again, giving you more out of your sequencing run. So I also want to take one quick minute to mention some of our exciting Agilent community designs. So these are NGS panels designed for specific use cases that leverage content expertise from some of the top researchers throughout the world, right? So these panels predate a lot of the improvements that I spoke about in the previous slides, but I think the value here is really the content itself. And as you can imagine, as we continue to add on to our portfolio of community designs, we can certainly continue to or actually start pulling in some of those innovations in future designs. But I want to talk a little bit more specifically about kind of the diversity with regards to the applications. Here is some examples that you'll see, including the Glasgow Cancer panels, which work in partnership with the amazing folks at GPOL. We have some infectious disease panels, such as the SARS-CoV-2 design from experts at the University College of London. And then just a number of panels focused on model organisms for some of the more research folks, mouse, rat, and canine as well. And just before we move on, I do want to say with regards to the GPOL community designs, there's a lot of really great information about these designs available on our website. Probably don't have enough time to do a deep dive here, although it definitely warrants one. So I do encourage you to see, if you're interested in these panels, to talk to some of our team members, because these panels will not only allow you to detect SNPs and indels, but it also allows you to understand copy number variation, structural variants, TMB, MSI. I mean, I highly recommend speaking to one of our team members to really learn more about these panels, as they might be really impactful in your lab. Finally, if you are new to the process and need a little bit of extra help, or if you perhaps are an experienced user, but you're working with a particularly difficult region, I highly recommend our custom design testing service, which can certainly help you. As I mentioned, we've been doing this for over a decade, and you'd be able to essentially leverage all that expertise and domain knowledge to get yourself up and running in typically about 6 weeks. I could not recommend working with the DTS team enough. They are an excellent group to work with, and very, very great service, especially, again, if you're new to this space. So kind of following along that, I did want to provide some data comparing some first pass designs compared to the optimized designs that you would get from our DTS team. And I'm using a small cancer research panel about 1 megabase as this example. And here you can see that the optimized, which is in blue, was able to decrease the fold 80 penalty. It was able to improve both the coverage depth and the mean target coverage. And this is all compared to first pass. So again, a really good aid in getting into targeted enrichment. Okay, so we've covered a lot of grounds in terms of targeted enrichment, and that's part of the overall NGS workflow, where Agilent is really a foundational member of the NGS community. But even the best targeted enrichment platform still doesn't work in a vacuum. You need to surround it with the tools that make sense. So I'm going to shift gears now to our library prep portfolio, where we've also strived for continuous improvements. Okay, so I know there's a lot on this slide, so I'll unpack this piece by piece. First off, I want to highlight this timeline shown on the top of the slide. And as you can see, Agilent has been investing and innovating in library prep, NGS library prep, for a very long time. Along the way, we've addressed key needs of the rapidly evolving NGS community, making it possible to access an even broader set of samples from a variety of input material, right? If you turn your attention to the table on the bottom, though, you'll see that specifically where our library prep capabilities have changed to keep pace with the various sequencing technologies. This includes interrogation of unique dual indexes, to help solve some of the index hopping issues which may be observed in sequencers using pattern flow cell and exclusion amplification, and molecular barcodes, some know it as UMIs, which make it possible to differentiate unique library fragments from PCR duplicates, which could be an issue for certain applications, and we'll talk about that in a later slide. You can also see where we introduced pre-capture pooling to help make larger sample number projects more financially realistic, which is fueling much of the drive that we've seen towards really remarkable undertakings like gene atlasing and even population sequencing. And to make this all even a little bit more palatable, today I'm going to focus specifically on the newest library prep options shown here on the right in blue. These are the SureSelect XCHS2 DNA and SureSelect XCHS2 RNA. Okay, so let's dive into the DNA version of SureSelect XCHS2 first. And you can see I've dropped a copy of some of the key characteristics and features on the bottom right-hand corner. This will make it kind of easier to keep things straight as we go along. One of the most important innovations Agilent has ever made in the field of library prep actually relates back to the targeted enrichment. And that's the ability to accelerate the hybridization of biotinylated RNA baits to DNA NGS library fragments. Historically, getting efficient hybridization enabled to pull those fragments out of solution and sequence them required an overnight incubation at a very specific temperature. But by refining the library prep process, Agilent has brought that hybridization time down to only about 90 minutes. So what this robust yet accelerated hybridization time really means is that you can now do this in a single workflow. So here I'm showing two different options. Of course, you could take your time and really spread this out throughout the day, spilling over to day number 2 to do capture and wash. There's a safe stopping point, which will require you to put your samples in 21 degrees Celsius hold overnight. Or if you're having a really ambitious day and you have 9 hours allocated in your day, you can certainly get through it within that day and go through all the way to PCR amplification. So I also wanted to mention that another very cool aspect of the XDHS2 DNA is that it can actually be used to produce libraries. It can actually be used to produce libraries that can be used for either whole genome sequencing or targeted enrichment. And you can see over here on the left hand slide, there is a real simplified representation of the library prep workflow. If you do all 4 phases, including targeted enrichment, you'll end up with enriched libraries that target a very specific subset of the genome, as I mentioned previously. Alternatively, if you forego the enrichment step and stopped after the final PCR amplification shown here, you can go through to whole genome sequencing. So more and more, we're seeing a trend towards a multi-omics, multimodal sequencing-based approach. And you can imagine the benefits of using a consistent library prep across both genome and exome runs for each sample. I'll also take a moment here to mention that XTHS2 has shown major promise in being able to eliminate the final PCR step altogether. So we're looking forward towards supporting a PCR whole genome sequencing workflow soon. So now I want to begin transitioning to the SureSelect XTHS2 RNA portion rather than the DNA. And I really want to emphasize a remarkable, exciting aspect of these two kits. So the XTHS2 RNA is built around a common backbone shared with XTHS2 DNA, meaning that once you're done with shearing on the DNA side, and once you're finished cDNA synthesis on the RNA side, you're able to process these samples within a parallel workflow. So, if we circle back to the emerging trends towards multi-omics, you can imagine a scenario where you start with one sample and use a single highly parallel workflow to end up with 3 integratable data sets. That's whole genome, targeted DNA, and either whole or targeted transcriptomes. And these data sets can be analyzed together to achieve a deeper biological understanding, of course, than would be possible if you were to just use one of these approaches. And whenever you add in the fully supported native automation from Agilent, either on the Bravo or the Magnus platforms, you have access to an undeniably robust and powerful NGS workflow. So I want to highlight one more aspect of our RNA libraries, which is our addition of the molecular barcodes, or MBCs, UMIs, if you'd like. But first, let's take a step back to actually see how these really enable a very particular application, right? So many folks that are interested in RNA-seq typically, targeted or otherwise, typically focus on counting applications such as gene expression, and a number of others. Now, when you are preparing your libraries, and more specifically PCR amplifying your library prep product, you often deal with PCR duplicates, which can sometimes throw your count off, which is demonstrated in that first graph. If you choose to ignore the fact that you have PCR duplicates in your final data set, you will likely overestimate the count. And as demonstrated in that first graph, you can see that you might be counting certain genes a little bit more than what's really there. So one way of compensating for that is to perform deduplication techniques, such as eliminating reads that are perfectly overlapping. The idea being that statistically they should probably be a little bit more random than that. However, this approach tends to now underestimate the count, as you'll likely throw away reads that are unique and not duplicates. So with the addition of these MBCs or UMIs, which provide unique sequences on each one of these library preps, you can see that here in the orange, you're now able to specifically distinguish between two reads and determine if they're duplicates or not, right? So this plus potentially some dedupe techniques that I spoke about before really allow you to get the most accurate count, which you can see in this final graph here. So I want to turn now the discussion to an often overlooked aspect of the NGS workflow, and it's particularly relevant as these technologies continue to make their way into a more clinical setting, and that's automation. So Agilent has a long history of being a trusted provider of lab automation of all shapes and sizes, and it's likely you're very familiar with our incredibly well-adopted and much-loved Bravo platform. And Bravo is absolutely a workhorse within the lab, but I'm going to not focus too much on the Bravo. Instead, I want to focus on something a little bit more new and talk a little bit more about the Magnus instrument, which you can see here. So just a couple of highlights for Magnus. This is a compact NGS prep system designed to automate and vastly simplify the library prep and enrichment workflows. So at a push of a button, Magnus can autonomously complete all steps of the library prep and enrichment for up to 8 samples in as little as 9 hours. And please note that this Magnus system has been designed from the ground up to really meet the needs of these clinical labs, and it's already earned the CE-IVD classification as well too. So really have the clinical space in mind. So when I say Magnus operates autonomously, I mean true walkaway automation. You don't have to come back to move plates or fill bottles. There's no adding tips or transferring to a thermocycler. No human interaction needed after you push that button, except to, of course, pull your samples back out. The system is efficiently designed such that it reduces lab footprint. And of course, this is important because lab bench space is precious real estate that we want to really carefully monitor. It also includes prepackaged and fully sealed reagents that are keyed and barcoded, so really kind of mistake-free loading here. There's even an onboard UV lamp that ensures decontamination between runs, which is really important for the busy lab. And we really paid attention to the details here to make sure that every user, whether it be research, clinical, or translational, can have confidence in using the Magnus. So in addition to that, we also have our very precise thermocycler that's also on board to do the final library amplification process. It also applies a single-use seal to the top of the plate, so you can store these libraries up to 72 hours. That means when you push the start button, you can walk away and don't have this urge to rush back to the instrument to pull your samples off. And this includes even going over the weekend as well too. So really important for those folks looking forward to the weekend. I also want to call out the fact that the Magnus includes an onboard wizard, which helps walk you through that process with images and really clear instructions. So really with minimal training, almost anybody can elevate their lab efficiency using the Magnus system. One last point here with regards to the Magnus, and we love our manual workflow on SureSelect. It's a relatively quick and easy workflow with about 2.5 hours of hands-on time, but the Magnus is actually able to bring this down to just 5 minutes, right? All that loading time to essentially get your samples up and running. So again, another huge advantage for folks that are just looking for a little bit more lab efficiency. Okay, all right, so that has been quite a journey through the whole NGS portfolio. Of course, 20 minutes is just really not enough time to talk about everything. We still have the QC portion of the portfolio, the informatics portion of the portfolio. So I do encourage you to go out to your Agilent reps, have a conversation, and really kind of see how we might be able to connect all these pieces to get your lab running efficiently. Before I jump to the questions, I'd just like to thank you all again for choosing to spend your time with us today. It's been fun going over this portfolio. So maybe I'll pause there, David, for some questions.